Bismillahirrahmanirrahim. This video follows the video regarding grossing of a specimen, and in this we are going to discuss what is known as tissue processing. Tissue processing. Now, what is tissue processing? Tissue processing is the technique by which the tissue material taken in the cassette can be cut into thin slices of 2 to 4 micron thick. Now, it is impossible to cut big biopsy tissues into such thin slices by a knife. So what does tissue processes involve? Tissue process involves making the tissue into a paraffin block, what is also known as a tissue block. And the paraffin block can be cut into thin slices by a machine which is known as microtome. Now, in the last grossing, we ended up with the showing you up with the cassette over here, and this contains specimen taken from biopsy, specimen taken from grossing. In the grossing, we had looked at the gross structure of the organs which were submitted in the lab, and we had taken biopsy from the suspicious part of the specimen, and these were kept, small pieces were kept in these different cassettes, and they are all labeled with the patient's file name and with the location of the tissues taken from there. So from here the tissue processing will begin. Now, what is the aim of the tissue processing? The aim of the tissue processing is to make very small film from big structures from tissue sample. Now, the tissues are sent in big jars, big specimen, and what is your aim is to make a thin slice, almost 2 to 4 micron thick, which can be seen under microscope. Now, what does it involve? These are big chunks of tissues, and these are 2 to 4 micron thick. How do we go from big chunk of tissues to such thin film slides which can be seen under microscope? So that is known as tissue processing, and this involves a number of processes, a number of stages, which we'll discuss one by one. So important thing number one is, whenever you get a tissue from the any material from the physician or from the surgeons, important thing is number one, formalin. Put the tissues in formalin, 10% formalin, so that it is fixed and it is preserved. So that is very important. You must receive the specimen in formalin from the operation theatre, from a surgeon or from a physician who has submitted say an endoscopic biopsy, and you must keep it in formalin so that the tissue is preserved. If this is not done, the tissue will get petrified and you won't be able to do any processing. So that is very important, formalin, keeping the tissues in formalin. Now, once you have done that, then the steps of tissue processing are: number one, dehydration; number two, cleaning; number three, embedding; and number four, sectioning. Now, I'll discuss one by one. What does it involve? Now, once you get the tissues from the specimen which you cut, important thing is those tissue contains water, and your aim is to have paraffin inside the cell. Replace the water with paraffin inside the cell. So you can make paraffin blocks, and then the paraffin block can be cut into finer slices. So first stage of you can't infiltrate paraffin directly inside the cell because it contains water. Paraffin is not soluble inside the water. So important thing is removing the water from the cells, and that is known as dehydration. Now how can you remove water from the cells? Now that's not very difficult, that's very easy. By putting them in different solutions of alcohol. So what you do is you start with 70% alcohol and go towards 100% alcohol. And you put your cassette in 70% alcohol from 8 to 9 am. Now this is the tissue processing. From 8 to 9 am, each is for 1 hour. Then from 70%, the increase the strength of alcohol to 80%, then 95%, then 95%, again 95%, and go up to the 100%. So from 8 to 2 o'clock, about 4 and 2, 6 hours, this is dehydration. This is dehydration. Dehydration. What you are doing is removing the water from the tissue, from the cells. So once it is dehydrated, all the water has been removed from the cell. But you can't still infiltrate paraffin inside the cell, because paraffin will not go into the cells unless it finds something which is where it is soluble. So after dehydration, now this is the process of dehydration. We do it manually. We are not so much advanced, and we do not have so much of expensive equipment. So what we do it is manually. So that is formalin to preserve tissues, and these are different jars. This jar contains 70%, this alcohol, this contains 80%, that is about 95%, 95%. So the cassettes, here you can see cassettes placed in formalin to preserve the tissues and to get it fixed. These cassettes will be then transferred for 1 hour into 70% alcohol, another 1 hour for in 80% alcohol, in the next 1 hour in 95% alcohol, and then 95%, and this will go into the next 100% alcohol, and this will take time for it to dehydrate. Now once the cells have been completely dehydrated, you have the second process, which is known as clearing. Clearing is removing the alcohol from the cells now. Once the alcohol has gone into the cell and the water has come out of the cell, you have to now remove the alcohol from the cell. Or how do you do this? Now this is done by xylene. This is the process of clearing. Xylene you keep for 1 hour and for 2 hours in xylene. So after dehydration, you want to remove alcohol from the cell. The xylene will replace the alcohol now. Now this is also done manually. That's 100% alcohol, and these are the 2 jars of xylene. So they are kept, the cases are kept here for 1 hour. This is also done manually. Now once you have done this, completed the clearing, so this has taken 6 hours, another 2 hours, that is about 8 hours. Then you put them, then you are ready. Once the xylene has entered the cell, you can introduce paraffin inside the cell, because paraffin is soluble in xylene. Paraffin is not soluble in alcohol. Paraffin is not soluble in water. That's why this process is done. You have to dehydrate first to remove water. Water will be replaced by alcohol, but you can't infiltrate paraffin in alcohol because paraffin is not soluble in alcohol. Then you replace alcohol with xylene, and once the xylene is there, then you can put paraffin wax inside, because paraffin, liquid paraffin is soluble in xylene. So that is known as clearing. This is clearing. That is clearing. Now we come to the next stage, which is embedding. Which is embedding. Embedding is embedding liquid paraffin inside the cell, which will replace the xylene now. So what we do is, once we have got over here cassettes over here which has gone through dehydration and clearing, contains xylene, you put liquid paraffin, hot liquid paraffin, inside the in the cassette, so that the liquid paraffin now goes into the and replaces xylene. And at the end of it, once you have done that, you need to cool it, put the lid on, and this is known as paraffin block or tissue block. This is known as paraffin block or tissue block. And this is the final appearance of the paraffin block or tissue block. So what we have done is, we have taken the biopsy material, put it into a cassette from the after doing grossing, and we have passed it through dehydration, clearing and embedding, and now this has become a paraffin block, and this paraffin block can now be cut into smaller, very thin film of 2 to 4 micron, and this is done by a means of microtome. This is a machine which is known as microtome. Microtome machine. And once you put the paraffin block inside and you make smaller slices, these are the slices coming out. And once you got the slices, these thin sections are put on the water bath. This is the thin section put on the water bath, and this will rehydrate. So the process of reverse process now starts. From dehydration, the cells will again get water inside, and these thin sections are put in the water bath, and then they are lifted up with a glass slide, and you got it over here. This is the thin slide. Now these thin slides need to be stained. The next stage will be staining. So staining, I'll write it down over here. Staining is done by 2 dyes. We've got 2 main dyes. One is hematoxylin and the other one is eosine. Hematoxylin is blue in color and it is basic in nature. So when the slide is put into hematoxylin, all the structures which are acidic in nature will take up the basic dye, which is hematoxylin. For instance, nuclei contains nucleic acid, so it will take up hematoxylin, and the nuclei will become bluish in color, purple bluish in color. Another dye is eosine. Eosine is acidic in nature and it is pink in color. And all the basic structures will take up acidic dye, which is eosine, which is pink in color. So the cell membrane, cytoplasm, they contain basic structures, so they will take up eosine and they will become pink in color. So once the staining is done with eosine and hematoxylin, the slide is ready for inspection under microscope. So this is a teaching microscope where the teacher can, another person can see over here what is seen inside. So that's the tissue processing. So to sum up tissue processing, once again it contains a number of processes, a number of techniques which needs to be, you should be skillful and you should be aware of the reasons for it, doing it. In our parts of the world this is done manually because we do not have so much of budget to get a very sophisticated machine. So we have kept it simple, but we are all the time improving upon it. So the steps of processing are: number one is formalization to preserve tissues. Next is dehydration, removing the water from the cells by means of alcohol. Once the alcohol has been infiltrated into the cells, you have to clear it because still paraffin cannot go into the cells. So the clearing is done by xylene. The xylene will replace the alcohol, and once the xylene is replaced the alcohol, you can introduce liquid paraffin. That is known as embedding. And the last is, once you have got the paraffin block now can be cut into very thin slices, sectioning by means of microscope. Thank you. I'll stop over here. And I hope you find this useful. Thank you.